to get a general show of hands here. Who has studied biology or biochemistry or anything like that throughout the time here? All right. Got a good show of hands here, and a few who haven't. And so this, for a lot of you, this may come across as review, but I just like to set up preamble to the systems in our body that are affected by drugs. That being said, any system in your body can be affected by drugs, but I'd like to frame the question of drug interactions as A to B to C, and how one effect will lead to another effect, to a final effect, regardless of what specific drug we're talking about. So I'd first like to talk about signal transduction. That is the way that signals or information is passed from cell to cell. One way is nerve signaling, and that's of course what takes place in our nervous system, central and peripheral nervous system. And in this mode of signal transduction, signal is carried in an electrical wave along the body of a nerve cell. And so it's an electrical impulse that can go all the way from a point of origin right to the terminal area. There's also chemical signaling, and so that's where cells communicate to adjacent cells through a chemical intermediate. That's what carries the information. And so in the example I just showed you of nervous signaling, at the end there's the tip of a neuron, and neurons communicate between each other with this chemical signaling mechanism. There's hormonal signaling, which is a form of chemical signaling, but it's more about multicellular organisms or when communicating from one area of the body to another. I've got a few examples here. Estrogen, testosterone, synthesized by the body in one place for final activity. And lastly, there is physical signaling. Physical signaling is really any start of an information pathway that comes from a mechanical stimulus. So 3 of our senses are actually based on hearing. So in the case of hearing, sound waves pass over this area inside the eardrum, or inside the ear here, and causes it to vibrate, which stretches these little hairs here and turns that into the electrical signal, and then now we're back into nervous and chemical transitions. And very similar with touching. Small sensors under the skin that are called baroreceptors, and they're sensitive to pressure. And once the pressure is applied, they begin to activate and send signals to the brain. And sight. And so I know I said mechanical stimulus, we'll call light also mechanical stimulus. And it's a specific process, but light impacts the retina here and is transformed into an electrical signal, which is then decoded in the brain to form an image. Okay, so that's how cells talk to each other. Now, when a cell is sending a message to the other cell, how does the other cell receive it? And it does that through transmembrane receptors. So people here know that cells have membranes, and that proteins can span these membranes. In the case of a receptor, the chemical can interact with one end on the outside of the cell and induce a change in the shape of the protein, which will be conveyed all the way to the other side and pass it into the cell. Now, there's 2 main divisions of that type of interaction. There's the kind when your chemical, called ligand in this case, binds to a receptor, and as mentioned earlier, the change in shape will cause a change in activity on the other side of the cell, on the inside of the cell. That can mean lots of things. It can catalyze a reaction. This transmembrane protein could couple to another transmembrane protein of the same type after that. In any case, there's always there can be many downstream effects. The second major kind is when the binding of the ligand, when it binds to the protein, it opens it up and actually makes the cell, primitivezes the cell with respect to another chemical. And so in the case of nervous depolarizations, you see that everywhere. You see that these channels are changing the electric potentials on either side of the cell, allowing for depolarization to take place. In the case of psychoactive compounds, that's going to be a predominant theme, is changing how and when cells polarize. A little brief bit on metabolism, and this will be fairly familiar. There are metabolites, and those are the small molecules within your body. The exact definition isn't precise, but for the sake of what we're talking about, I'd like to say that it's small compounds in the body that aren't involved in, aren't genes, aren't involved in any structural functions. We'll say intermediates in metabolism. And in between the metabolites, there are enzymes that change one metabolite to another metabolite. So you see in the case of a lot of drugs, the form that you ingest or take the drug in is not the active form. It's converted in the body, and that'll be done by enzymes. So you convert it to its final form. Here's an example within the body of how that changes. So the teal dots are your metabolites, and the pink dots are your enzymes. And so this pathway starts with phenylalanine, an amino acid that we synthesize in bulk in the body, and then through more and more chemical changes catalyzed by the enzymes, we work our way up to dopamine, epinephrine neurotransmitters. And so that's how something like an amino acid can be changed into a more specific molecule for signaling. And then just to point out the equilibrium and feedback properties of this all of the metabolic pathways in our body are in a state of equilibrium with respect to one another. And so if you induce a change at any one point, it's likely to affect surrounding points in a metabolic pathway. You change one thing, you cannot help that it's going to change others because they're all interconnected. And then when talking about feedback, that means that sometimes a small amount of one drug, something that's introduced, or a small amount of something within the body, in the case of hormones, can be amplified, because we're talking about a signal moving from point A to point B. The exact chemical intermediate, I'm trying to downplay in all this, because the signal can be initiated in a very small location and be amplified through various chemical intermediates, or through the nervous transmission. One neuron can connect to many and amplify a signal. Okay, drugs. Okay, so I guess a drug can be any substance which you introduce into your body that wasn't there before, that changes its systems. And so I've gone over and talked about enzymes and metabolites. A drug can be either. It can be anything really that affects one of these systems in your body. Usually it's a small chemical level. I was thinking about earlier today, thinking about there are no nervous drugs, but I thought, well, a pacemaker. Put a pacemaker in, it's there. It's introducing an effect that wasn't there before. It's returning you to a normal function. And so even though when you take a pacemaker, it's doing the same thing. So I'm trying to dissolve those lines. Okay, so I've got a few examples to talk about all this. Acetaminophen, Tylenol. It's a pretty direct example, and so after you take the Tylenol, it will work its way into your body, and it's actually an inhibitor of cyclooxygenase 2. It's an enzyme that normally will take arachidonic acid in your body and convert it to this compound here, prostaglandin H2, and that's an inflammatory compound. So as that moves on down through different pathways, it'll lead to tissue inflammation. And so when you take acetaminophen, it actually blocks the enzyme from doing its function, and you're lowering its effect. And so that's a drug that affects the body more so than the mind. And now here's a drug that affects the mind to quite an extent, caffeine. So it's an endosine receptor antagonist. I'm sure many of us have heard it, but just to say it again, antagonist means it has the opposite effect of what it's affecting, or the opposite effect of its natural effect. An agonist has the same effect. So there's a caffeine molecule, and here's an adenosine molecule. And so you can see the structural homology between them. And that's usually the base part of designing a drug, is finding a way to mimic but in a different way, the natural metabolite. Okay, so adenosine, normally it binds to the receptors in the brain, releasing dopamine, or in the body it suppresses heart rate. And so when you take an antagonist like caffeine to adenosine, you find the opposite effects take place. And so caffeine could induce, has been shown to induce dopamine release, and that's sort of tentative the way the research is going. And it may be associated with the sleep-wake cycle. And so there's many there's much anecdotal evidence that I'm sure we could all relate to, thinking about drinking too much coffee and screwing up your sleep cycle. That's something we all know, and now the biochemistry is it's only now being elucidated. But morphine. Morphine, I wanted to talk about because it sort of involves a lot of systems that I've talked about. So it's an opioid receptor agonist, and so it affects the opioid receptors in our body like our natural metabolites. It's actually named after it, after opium. And so in this case, though, morphine, it does have effects on its own, but in terms of pain relief, morphine is ingested or metabolized in the liver to these two compounds, morphine 3-glucuronide and morphine 6-glucuronide, and a few studies have shown that morphine 6-glucuronide is the one that causes pain relief. When talking about recreational use of opioids, ingestion is not the primary path, and that's because simple pain relief is not the primary goal. And if you look into recreational use, you'll see that the opposite effects are true. To avoid liver metabolism is the goal. All right, and I just got a few thoughts on all this. I guess I want to talk about, I don't know if genetic interplay is the right word, or right few words, but if we've got our individual genomes, we've got many of the same genes, but we have different expression levels perhaps, and we're all a unique, we're all a unique individual. But in terms of how drugs affect the body, it seems to me at least that to introduce a substance into your body that will arrive in your body as a metabolite is the same as having a genetic pathway in your body for the time being. I consider morphine. We make morphine ourselves, but in small amounts. But having a drug that we don't make on our own, it's as though you've given yourself that biosynthetic pathway. And then, there are consequences from that. And also I'd like to note the subjectivity of experience with any and all drugs, pharmaceutical, recreational, or what have you. Because we're unique genetically, we have different enzyme levels. There may be 7 or 8 conversions in the pathway when you take a drug, how it affects you, and we have slightly different levels of all of these enzymes or enzyme activities. And the effects can be very different. So when it comes to developing new drugs medically, they do a lot of testing and they find out maybe which population subtype it works on, and investigate that. In the recreational sense, not so much. It's a lot of information being passed from friend to friend, saying how something went or something along those lines. And so perhaps good to keep in mind that we are all unique and we use a lot of things differently. So there's an experiences are something. I'd like to thank Wikipedia, Google, the Library of Science, and this gentleman, Robert McCarley, who did the research on caffeine and dopamine. That was something specific. I should put a name up. Thank you very much.